Welcome, SACNistas. Thank you for joining our session, Many Hands Make Work More Interesting. In this session, you're going to hear from 4 researchers involved in highly collaborative, multidisciplinary research projects down in Antarctica. 3 of us are biologists and 1 of us is a glaciologist. We organized this session because we want to tell you about the unique training opportunities associated with our research, and we're hoping you'll join our team either as an RU or as a graduate student. At the end of the recorded session, we'll be joining you live to answer any questions you might have, and we hope you'll stick around to meet us. Our first talk today is by me, Christina Takis-Vesbach, and I'm going to tell you about the McMurdo Dry Valley Long-Term Ecological Research Project. Our team is led by Mike Gusev at the University of Colorado and includes 8 co-investigators who are located at universities across the U.S. We have a big team. More than 40 people are actively involved in the project, including undergrads, graduate students, postdocs, and collaborators. Our project is one of 28 sites that are funded by the LTR program, which was initiated in 1980 by the National Science Foundation. It was started because they recognized that it's impossible to track and understand ecosystem variability and responses to environmental change with the standard 3-year grant. LTRs host large teams of scientists asking fundamental questions about how ecosystems work. We maintain long-term observational records and conduct large-scale experiments. Sites include aquatic and terrestrial sites, agricultural and pristine systems, and even the human environment. Each grant lasts 6 years and has the potential to be renewed multiple times. Some grants have been funded for 40 years, while some new sites have been added in the last few years. Our project's been funded since 1993 and had field seasons every year until this year. Sites are located across the U.S., from the tropics to the Arctic, and there are 2 sites in the Antarctic. Our site, McMurdo, is a terrestrial site, and the Palmer site, which is on the peninsula, is a marine site. You'll hear more about the Palmer LTR later in this session when Jeff Bowman gives his talk. The McMurdo Dry Valleys are by the Ross Sea, off the western coast of the McMurdo Sound. They include an area of about 4,800 square kilometers. Our site is the coldest, driest site in all of the LTR. The annual temperature is minus 20 degrees Celsius, and the area receives less than 20 millimeters water equivalent of moisture most years. All of this is in the form of snow. It doesn't rain here, so it's a very dry environment. The ecosystem includes 4 main habitats. A majority of the area is covered by soils, which, as you can see, host no plants. There are also streams, which are fed by the glaciers, but they only run in the summer, when temperatures rise above freezing. It's dark all winter long, so there's no stream flow then. This runoff flows into the lakes in the valleys, which are mostly closed basin or have no outflow. The lakes are covered by up to 5 meters of ice year-round and maintain liquid water columns beneath. Despite the harsh environment, life abounds, but it's all microscopic. There are bacteria, archaea, and eukaryotic microorganisms and microinvertebrates in every habitat. At the base of the food web are phototrophic bacteria and algae, and nematodes, rotifers, and tardigrades are at the top. There are even diverse viruses that we're just beginning to investigate. The microbially dominated ecosystem enables us to address fundamental ecological questions without the confounding factors posed by plants and animals. We're currently focused on understanding landscape connectivity. We became interested in this because during the first decade of our project, the local climate was cooling, and we know that in cold years, the ecosystem was on the verge of being uncoupled. There would be little glacial melt, the streams would barely flow, and consequently lake levels decrease during this time. The habitats in this ecosystem are connected by water, and if streams don't flow, there's a lack of connectivity. But then in 2001, we suddenly had a massive flood, and the ecosystem has never been the same. We now understand that the flood happened that year because of a large-scale climatic anomaly that led to clear skies and more than 50 days of above freezing temperatures that summer. This is a picture of the Onyx River under normal flow and then during the flood. After the flood, we saw major changes in many aspects of the ecosystem. For example, the lakes became very turbid at first, which depressed primary production, but then, within the next couple of years, it was stimulated because of the massive influx of new nutrients into the lake. We also observed changes in the soil communities and many of the physical and chemical characteristics of the ecosystem that we measured. After the flood, it seemed that the ecosystem had undergone a state change. Air temperatures and solar radiation continued to climb for a number of years, and lake levels rose. And we thought the rapid warming that had been happening at the peninsula for several decades had now arrived to the McMurdo Dry Valleys. Although now that we've accumulated a long enough temperature record, we can see that since 2006, temperatures have actually been variable and not continued to rise. However, we still are seeing lake levels rise because of thawing. The glaciers continue to melt, mostly because of clear skies and the deposition of sediment on their surfaces, which leads to more warming and more melt. This led us to our current overarching hypothesis, that is, increased ecological connectivity will amplify the exchange of biota, energy, and matter, homogenizing ecosystem structure and function. We use a combination of monitoring and experiments to address our hypothesis, and this effort requires more than just biologists. It requires scientists from different fields. Let's hear from some of the team members about our approach.
The LTER project is an interdisciplinary project because it has a lot of different people coming together to answer really big questions. When we write our proposals, our scientific proposals, we lay out hypotheses that none of us could answer by ourselves or none of us could address by ourselves.
As a grad student, I can say working with the McMurdo LTER gives us opportunity to engage in interdisciplinary research alongside scientists from various fields. Collaborating with others and making connections like these is unique in graduate education, in my opinion. I feel extremely lucky to gain experience and understanding of research fields outside of my own, and I imagine these experiences will help me in the future when I'm applying for jobs after my PhD. So having great connections like these, meeting different people, and also in terms of field work, running just such a large field campaign. So the logistics of, getting out into the field, taking helicopters, getting all of your equipment ready, because once you're out there, and if you've forgotten something, then too bad. So really planning is a huge aspect here, and working with a bunch of different people from different backgrounds is a really great experience and unique in my opinion.
Working in Antarctica is spectacular, and the privilege to travel to this continent is not lost on us. But one of the most rewarding aspects of this project is the team that we get to work with. Many of us have worked together for decades now, and the fact that we live in the field together for weeks to months on end has really brought us together.
And then I think the interdisciplinary nature of this work requires a real team-focused approach to science. So that is seeing the project for your own scientific goals, seeing how those scientific goals fit into the overarching objective of the entire project, and then at times putting the project ahead of your own individual interests.
Also, while in the field, we get to live with our students, and that's also another great opportunity for both us and the students to become closer and to work closely together. But it's not all work. Camp life is fun, and the camaraderie that develops in camp is one of the rewarding aspects of the project too. If you're having a hard time envisioning yourself working down in Antarctica because maybe you don't feel like you're outdoorsy enough, and these pictures look like everyone knows what they're doing, and they are comfortable in the cold and working outdoors 24/7. Many of us come to this project with little outdoor experience, and there's plenty of training and plenty of warm clothes. So don't let the remoteness or the foreignness of this ecosystem deter you from working with us, especially if you think you would really like to do this kind of work. We currently have opportunities for both undergrads and grad students to join our team. Next summer, we'll be hosting an REU program for undergraduates. It'll be stateside, and you'll have the choice of joining any one of our labs, and the application and more information is available on our website. Grad students can apply directly to different PIs. Take a look at our website, identify the kind of research you want to do, and reach out to one of us. We'd be happy to talk to you. I want to acknowledge our support from the National Science Foundation and Antarctic Support Associates, which provides us all the logistical support that we need down in the field. And of course, all the great team members of the McMurdo LTR and my co-PIs. And of course, SACNAS for giving us this opportunity to tell you about our project. Thanks.
Hi, my name is Helen Fricker, and I wanted to talk to you about work that I do in Antarctica. And most of my work is actually involving looking at satellite data. And the reason why we need satellites to look at Antarctica is because Antarctica is large. It's just a massive continent, and really the only way that we can get a really good understanding of whether the ice sheet is growing or shrinking is if we can routinely monitor with satellites. And the instrument that I work with to do this is called an altimeter, and it just measures the height of the ice sheet. And it does that again and again. And over time, it builds up a time series of height measurements, which we turn into volume. So we can look and see the shape of Antarctica, and is it growing or shrinking? And if it's growing or shrinking, it tells us that it's either losing too much mass or it's gaining too much mass. It's just like your bank account, the amount of money going in. You're either going to increase your balance or you're going to reduce your balance. So same with Antarctica, the mass budget of the ice sheet is very important, because if you're losing mass from an ice sheet, then you're actually adding that mass to the ocean and causing sea level rise, which is very important for society. We've been working on the ground in Antarctica for the best part of 100 years. Early days were sort of heroic age of exploration, Antarctic exploration, and you would have heard of Shackleton and some of the expeditions back in those days. So this timeline here shows 100 years of Antarctic science. This also shows you where satellites come in the picture. The satellites that I work with have only been around since the early 90s, but we've had satellites since about the 1970s. The other thing that you can see on this timeline is in the middle, 1969, about 50 or so years ago. This is when the first women were, a research team that went to Antarctica was made of all women. So this is how long we've had diverse field teams in terms of gender diversity, working to understand Antarctica. So it's quite short really in the sort of lifetime of Antarctica, but we're definitely changing, and our teams are becoming more and more balanced in terms of gender and also in terms of ethnicity as well. So yeah, this was 1969. It was quite a monumental achievement actually, particularly because the entire field team was made up of women, but it was hard to find any information out about it, and it turns out it's the same year that we landed on the moon. So I guess most of the media kind of went to that news, but for Antarctic science, this was a big deal. And here's the first time I went to Antarctica. It's actually the only time I went. I'll explain why. So here's me standing on Amory Ice Shelf next to this pole, and over on the left is the pole as it was when it was planted there or put in there to measure accumulation. And this was actually the year before I was born. So that was quite poignant for me to be on an ice shelf knowing that there's a pole there that's been there longer than I have lived. So we were there doing calibration for satellite altimetry. So this is the grid that we drove on the ice shelf. They're 10-kilometer squares. There's 120 kilometers here that we drove, and we have 24 of these squares. So we're actually we're essentially building up a ground truth data set that we could use to validate our satellite altimeter measurement, because we want to know that what we're measuring from space is accurate. So the GPS on the back of our skidoos here, we're measuring very accurately the height of the snow surface, so we could calibrate and see whether the measurements coming from the satellites were accurate or not. So one thing we can do with these data once we have it is we can make maps of height. And height, when you're on an ice shelf, because it's floating, can be turned into thickness. So this is a thickness grid for all of Antarctica here on the left. And this is Amory Ice Shelf here in East Antarctica. I actually used this map and was able to extract the amount of marine ice that's refrozen onto the base of the ice shelf, which is otherwise quite difficult to get at. And one of the things that this sparked off was a field program in the Australian Antarctic Division drilling at these holes here through the ice shelf. So these ice shelves are floating on the ocean, and you're drilling through several hundred meters of ice to get into the sub-ice cavity and take samples of the ocean and the ice on the way down, and also the seafloor on the bottom when you get there. So this is the kind of infrastructure that was required to drill, a hot water drill through the ice shelf. Quite a lot of excitement here, multiple years of field work that was sparked off because of this marine ice map that I made during my PhD in Australia. So taking just a little bit further chunk of the ice sheet now, like not just the ice shelves, but going a little bit upstream as well, to show how we can monitor the ice sheet as a whole. Various different techniques we can use, but I'm still going to focus on altimetry, different types of altimetry now. So laser altimetry. I came to the States to work on a NASA mission called ISAT. And one of the things that we were able to find using ISAT was very subtle signals on the surface of the ice sheet that turns out were because of subglacial water moving in between the ice and the bedrock, moving away from one reservoir to another, and the surface deformation that we got as a result of that was detectable by satellites. Okay, so this is going to show you what that means. So this is coming through the ice sheet, this slice through the ice sheet. You can see water's going to go from the left to the right, and the surface of the ice will change as well. And that's what you can detect from satellites, which is amazing when you think about it, that 600 kilometers above the surface of the Earth, you've got an instrument that's able to measure something that's going on a kilometer or so beneath an ice sheet. Really amazing. And this was a really nice sort of discovery that happened in the late, it was 2007. And this sparked off another sort of field program, actually another drilling expedition to drill into two of these lakes. We actually drilled into this one here, Subglacial Lake Whillans, through the Wizard project. And then later on, Subglacial Lake Mercer with the Salsa project. So this is the kind of infrastructure that you need to drill into a subglacial lake. And in this case, we brought together a multidisciplinary team of biologists, oceanographers, and glaciologists, geophysicists to try and study this system as a whole. This is what it looks like at the bottom, the lake bed. This is actually at the grounding line, a fish that was found. This is the infrastructure that was required to drill into that lake on Whillans Ice Stream. Jill McCookie here, she's in this session as well. She was part of this project. It was just a delight to work with her. And this is watching the penetration of the lake on the ice. They actually were looking at it through camera. I was on text to my student Matt, who was taking part in this field work on my behalf. It was very exciting. I would get texts in the middle of the night telling me that they'd made it to the bottom of the lake. This is really cool stuff. So yeah, my other students have also been to Antarctica. Cecile is here, and Maya. And then this is Matt again, but this time in this picture, Matt is actually now an assistant professor at the Colorado School of Mines, and he is mentoring my student Philip, and they actually went down as part of the Salsa project just last year to kind of wrap things up. Now I want to look at the ice shelf again and just tell you a little bit about how the ice shelves are changing and why, using the altimetry measurements, a long time series now going back to the 90s, and we've managed to look to see where the ice sheets are changing. And it turns out that where the ice sheets are changing the most, which is essentially in these areas that are marked in red, this is where the ice shelves are thinning. Turns out that this is where the ocean temperature is warmer. So the temperature here in red is a warmer color, and the temperature here in blue is a colder color. So where you have the warmer water next to the ice shelves, that's where they're thinning the most. And you can see from this plot here that Antarctica is very connected in the global ocean. It's really, in the middle of everything. Even though it's at the bottom of the Earth, it's actually very, very central in terms of oceanography. So it turns out it's the ocean that's causing the changes in the ice sheet, which means, again, we need a multidisciplinary team to try and understand this. So along comes another project called Rosetta. This is a collaborative project. with a very large group of people looking at the Ross Ice Shelf, which is the largest ice shelf in Antarctica, the size of Spain. This is my student Maya again. And she was on board the C-130, where we launched these floats into the ocean to sample the water offshore of the Ross Ice Shelf. And this is because we need to collect measurements in these areas to figure out why the ice sheets are changing in these places and what are the oceanographic conditions in these areas that are leading to those changes. So this is just a little bit about me and my situation and why I only went to Antarctica that one time. Well, when I discovered the lakes, which was me in 2007, I was actually the mother of two young kids. So here, Zoe and Millie. And now, several years later, they've all grown up and we have another one. And basically, I'm a mother. So it's very difficult to go into the field. And so I've sort of done it from afar. I've managed teams from afar, and I've been at the end of phone calls and been super exciting. But I'm very grateful that I've been able to at least be part of it in some way. This is a decision I made. There are plenty of women that have been in Antarctica and really done a lot in the field. So yeah, I'm in there as well, but all these women here have just got such strong leadership in field work and other aspects of Antarctic science. And this map here is actually all the place names in Antarctica that have been named after women. So yeah, there's a lot of women to look up to and mentors and just leaders, strong leaders in this area. The appearance of Antarctic science from the outside definitely looks like this sort of heroic age feeling to it. The Antarctic explorer has definitely got that image. You know, this is Shackleton here. I'm thinking back to the early days of the picture I showed you at the beginning, but things are definitely changing in our community a lot. And diversity has become very important. The understanding that we really do need very diverse teams to understand systems, and working together as teams is definitely how collaborations and collaborative science is definitely the way to move forward our understanding in science. So definitely I feel like our culture is changing. This is a website I strongly encourage you to look at. And yeah, I just think this is a really great time to get involved in this field, and definitely very welcoming community that we are becoming. So the other thing is that it's very societally relevant what we do. And so speaking to the media is really important as well, and getting our message across in a clear way. So communication is vital, and actually talking to the media has become a very big part of what we do as scientists when we're working in Antarctic research. The other thing that we have to do, because of the whole link between ice sheets and sea level rise, is speaking to policymakers. And we have some real heroes in this area in our field. We have Richard Alley here, who testifies a lot before Congress. This is Twyla Moon and Robin Bell. Robin Bell is also president of AGU, and she was on an earlier slide. She's a real strong leader in Antarctic science. This over here is a Scripps delegation to COP25, and they met Nancy Pelosi. Cecile is here explaining sea level rise. I mean, there's just multiple ways that we can have an impact and get our message out there and make our work sort of heard in the broader area where it can really matter and affect policy. So just sort of thinking about it and bringing it all together, there's many things to definitely celebrate about what we've learned in Antarctica over the last 100 or so years. So it's time to sort of sit back. We have plenty of, I guess it's a community where we do have quite a lot of celebrations, but also we should be really careful and think about how we want to move forward with our work, contemplate like what do we want Antarctic science to look like in the future? How do we want our actions to sort of get out there into the policy world so that that actually will affect what the ice sheet will look like too because of climate change? And then communication, this is very important, like getting our message out to the public is vitally important. But I think the big one here is collaboration. Antarctica is large, it's a very, very vast continent. And we can really only make a dent in understanding it if we work in these multidisciplinary international teams, and not one nation is going to do this on their own. They definitely need to all band together. So this is very much where I sort of think Antarctic science is going in the future, these big sort of multidisciplinary international projects to really understand and drive our understanding forward of how the ice sheet works. So thank you very much. I hope you've enjoyed listening a little bit about Antarctic science, and feel free to contact me afterwards. Thank you.
Before I get into the talk, I wanted to introduce 3 students in my lab who have been moving our work in the coastal Antarctic forward. This is Tristy Desarathi, Beth Connors, and Natalia Arazo. So the Western Antarctic Peninsula is this part of the Antarctic continent here. It's the northernmost part of the Antarctic continent, extending out towards South America. And from the sea ice animation, you can see that the tip of the Antarctic Peninsula interacts very strongly with the sea ice edge. And this ends up being very important for the ecology of the region. Over here, we can see the climatology of chlorophyll, which is a good measure of biomass in the water. And here we have the spring, the summer, and the fall. You can see that the spring bloom starts out at the ice edge. And as that ice edge retreats back towards the continent, that spring bloom follows it. Now, the diatoms and the other phytoplankton that make up that spring bloom are really the key to the whole Antarctic food web. They support everything else in that food web. So here we've got the classic Antarctic food web. We've got the phytoplankton down here. They are coupled to the rest of the higher trophic levels, largely through krill. And those krill are fed upon by a whole variety of other really important species in the coastal Antarctic. The Palmer Long-Term Ecological Research Project is a long-term project that's been studying these ecological processes along the Western Antarctic Peninsula since about 1993. And the key part of the Palmer LTR project is a cruise that takes place every January on this vessel. This is the Lawrence Gould. And they go down and they spend about a month occupying different stations on this hydrographic grid here along the coast of the central part of the peninsula. On each hydrographic grid station, they measure temperature and salinity, they evaluate macronutrients, chlorophyll A concentrations, zooplankton community structure, primary production, bacterial production, a whole host of other ecological parameters. And they try to build up a long-term record of how these parameters are changing over time. Now, in addition to this expansive survey across the grid, there's also a small team that samples repeatedly at just a couple of sites at the southern end of Andres Island where Palmer Station is. This is a picture of that team out there on a very nice quiet day doing their sampling. Now, as a result of all of this really nice work, we have some understanding of how this ecosystem is changing over time. Apologies here for the mislabeled citation. So what you're looking at here is a very coarse look at primary production, or the amount of new carbon that's being taken up by the phytoplankton community, averaged across the entire grid that the Palmer LTR studies each year. We can see that it's very cyclical in nature. So there are years where it's low and then it increases, and then there are years where it's high, and then that process repeats. Now, these highs and lows are strongly coupled to different modes of climate variability, in particular what's called the southern annual mode. And the southern annual mode ends up being really important for the ecology of the region. This is a fairly well-documented climate phenomenon. Now, the southern annual mode really has two phases. There's a negative phase and a positive phase. And during the negative phase, ice conditions tend to be very high. Winds tend to be fairly low. This stabilizes the water column, allows the phytoplankton to grow. So you get lots of phytoplankton, and as a result, you get lots of krill, and it's good for the rest of the food web. During positive SAM conditions, the opposite occurs. There's relatively low ice. There's high winds. This results in a fairly unstable water column and a deep mixed layer that's poor for phytoplankton growth, and as a result, there's less food for krill and for the rest of the food web. Now, this all occurs because photosynthesis is dependent on light and nutrients, and these light nutrients are controlled by the wind and the sea ice. So this sort of establishes two extreme end members scenarios here. So you can imagine that if you're a phytoplankton cell and you're below this nice flat sea ice, you're protected from the wind, you're in a very quiescent water column. The conditions you're living in are very different than if you're under these conditions, where there's no ice, there's very strong winds, and you're being very constantly mixed down to those deeper, darker waters. Now, neither of these situations is particularly good for the growth of phytoplankton. The optimum is somewhere in the middle. You want a quiet water column, but you also want plenty of light to support growth. We wanted to understand how phytoplankton respond to the really rapid advance and retreat of sea ice in this region. We wanted to understand this on a very detailed physiological level. So to do this, we designed a microcosm experiment at Palmer Station in 2015. And this experiment was enabled by really unusual sea ice conditions. So here's a satellite image of the southern end of Andres Island. Palmer Station is right here. This is Arthur Harbor. And there's what we call landfast ice in Arthur Harbor in this particular year. So we've got nice ice that's anchored to the land. It's not going anywhere. It's flat and level, and it's easy to work on. This is fairly unusual conditions to be found in Arthur Harbor. Usually the sea ice is drifting in with the wind, and then it's drifting back out with the wind. So we were able to establish an ice station that we could repeatedly occupy and repeatedly sample from out on the ice. And at the same time, we were still able to go out to our regular sampling sites out at Station B and Station E by small boat. So these different sites represent different conditions. We've got underneath the ice out here at our ice station, we've got no wind and no light. But we also took water from that ice station, brought it back to Palmer Station, placed it in large transparent jugs, and put it in a circulating incubator here so it would stay at the right temperature, but it's screened to simulate 10 meters depth in the water column, which is a really nice depth for phytoplankton to grow at. So these phytoplankton have nice constant conditions, should be very good for growth. And at the same time, we're comparing those to more moderate conditions where we've got the actual natural marginal ice zone out here where there's moderate light and moderate wind throughout the season. Now, to do our analysis, do our comparison, we relied on a technique called metatranscriptomics. So we have 72 metatranscriptomes in our data set representing 24 different samples from these 3 different time points, as well as a variety of ecophysiological measurements, such as bacterial production and primary production, and even lipidomics, which is a technique that I'll talk about a little bit more later. Now, if you're not familiar with metatranscriptomics, very briefly, that's illustrated here by this figure. So you've got genes that are composed of DNA that are coding specific functions that are required by the cells to carry out some process. As the cells need to access that process, they transcribe that DNA into RNA. And then that RNA goes on and is translated into proteins. So if you look at the RNA as opposed to DNA, you're looking at what's actually being used, the genes that are actually being used by that cell in response to some kind of environmental condition. So we targeted that RNA component of the cell. And then we also looked at the ultimate end members of the enzymatic activity, which are these lipid components. So we're looking at kind of an intermediate process in that cell and the end member of that process. We hoped to develop a more complete understanding of the physiological response of the cells to the various conditions. This is just a quick look at the ecophysiological response of our samples to these different conditions. So here we have chlorophyll over time, here we have bacterial production over time, and we can see, for example, that in the at the ice station, which is represented by the circles here, there's very little increase in chlorophyll over their experimental period, whereas opposed to our mesocosms at Palmer Station, there's a fairly rapid increase in chlorophyll, and then there's a more delayed increase in chlorophyll out at the marginal ice zone. Bacterial production is a little bit different. Again, a fairly rapid increase in bacterial production. If you take that water from the ice station, you expose it to light. But even out at the ice station, there is a slow and steady increase in bacterial production as the season progresses. Now, what you're looking at here are kind of an initial look at the transcriptional response of different phytoplankton that are present in the sample to these different conditions. Basically, what we did is we took every sample in our analysis and we compared it to every other sample, and then we looked at what genes were differentially expressed between those 2 points. So it's a very naive and agnostic look at the data. We didn't really make any assumptions about how things would look. Now, the rows in these different heat maps represent different phytoplankton taxa that already have transcriptomes from experimental data sets present in a large database that we could access. So what we do is we take our metatranscriptomes and we map the individual reads in those metatranscriptomes to those reference transcriptomes. And it allows us to deconvolute the really messy mixed community metatranscriptome into discrete transcriptomes that represent specific phytoplankton. Now, over on this side, where it says response complexity, what we're looking at is the number of genes that are differentially expressed between 2 time points. And here, we're looking at the response strength between 2 time points. So complexity really relates to how many different genes are being expressed. So if you have a very complex response, physiological response to a set of conditions, you might express more genes as you're trying to alter your physiology to meet the new conditions. Whereas over here, you could simply have 1 or 2 genes that you are altering in terms of expression, but you're doing it a lot. You're really expressing that gene a lot. Now, in either case, there are a couple of specific phytoplankton taxa that really jump out as having a very strong response complexity or a very strong response strength. I've drawn up pictures here of those different phytoplankton taxa. So we have Thalassiocyra orchula, Corythron pennatum, and Fragilariopsis krugerulensis. These are 3 phytoplankton taxa that we would expect to find early in the season in this area. These are normal members of the WAP phytoplankton assemblage. So what you see in this plot here is values of what's called cosine squared. Cosine squared is a measure of the contribution of different genes. So we can see genes that are driving the response of Kerguelensis in green here versus genes that are driving the response in Corythrum penitum in orange and driving the response in Thalassiocera rotula in red. Now, if you look into what these genes actually do physiologically, you start to see that there's a lot of genes that are interacting with lipids, either that are involved in lipid synthesis or lipid degradation. So it really starts to become a story of lipid utilization within these cells early in the season. Now, lipids are tremendously important. This is a micrograph that's showing you the distribution of lipids within different diatom cells that are grown in culture. And the green, they're stained in such a way that the lipids are showing up as green. So here we can see these really abundant distributions of lipids within these cells. Lipids end up being a major source of carbon and energy for the rest of the food web. So what we find is that the environmental conditions really control the composition and the quantity of lipids within these different phytoplankton species. That has a huge impact on the quality and the quantity of the food source that's available to the rest of the food web. So with that, I would like to thank the collaborators on this project, including Hugh Ducklow, Scott Doney, Maria Kavanaugh, Ben Van Moy, Daniel Lowenstein, Helen Fredericks, Jamie Collins, Rebecca Gast, and Colleen Hansel, as well as the members of the Bowman Lab who continue to make really amazing contributions to our efforts in the Antarctic and in other environments that we're working at. Thank you very much for your time, and I look forward to taking any questions from you when we actually do the live session later in October.
Hi, I'm Jill McCookie. I'm a microbiologist at the University of Tennessee, and today I'm going to talk to you about how I explore subglacial environments in Antarctica, and it is highly collaborative work. So first I'd like to thank Tina Vesbach for organizing this session and for you all for attending. I'm excited to be a part of SACNAS this year. If you were able to look through the thick ice covers of Antarctica, you would see that it's peppered with hundreds of lakes, possibly thousands. There's saturated sediments, rivers, and streams that flow underneath this ice, right? And so it's this really exciting place that we have yet to explore. And it's truly a microbial continent. We have been able to tap into just a handful of subglacial lakes, and most of them have been in the Ross Sea region. So we've been able to drill into subglacial Lake Whillans and Mercer. This was only in the past decade. And we've also sampled a place known as Blood Falls, which I'll talk about more. And this is located in the McMurdo Dry Valleys, or over here in this region of Antarctica near McMurdo Station. So we've maybe sampled 3 over a continent the size of US and Mexico combined. So why is it so challenging? And one of the reasons is it's really difficult to get through thick glacial ice, but it's also really difficult to identify exactly where to go to target these systems and sample them. So exploring Antarctica's subglacial environment really does require many hands. And the way I think about it is, first you have to identify where it is you want to go to collect your sample and look for microbial habitat. And so this requires collaborating with geophysicists who can reveal where these lakes or saturated sediments might even actually be. And then you need to find a realistic target that you can access with a team or a crew to go and actually collect samples. And often these samples are below hundreds of meters of ice. So how do you get there? And at this stage, I like to collaborate with engineers who can help us reach these targets with novel tools. And then once you're able to collect samples, then you can start doing some microbiology. But even there, that's a broad collaboration, right? With chemists and physicists who can help you understand how microbial life is actually able to work in these exotic and extreme settings. And this type of research happens on many scales. So when you start with your geophysical tools, you're at the kilometer scale, which is like working across a city. But when you really want to know what a microbe is actually doing in its immediate surroundings, now you're down to the micrometer scale. So you're challenged not just by the interdisciplinarity of the work, but also on the many scales with which you're working. So why study these systems, and what is it that we're actually looking for? Well, subglacial ecosystems can take the form of these lakes that are trapped below the ice. There could also be saturated sediments, which we might think of as swamps or aquifers, There's the places where the ice hits the ocean, and you have this dynamic ecotone where you have sediment, ice, and water. And here's where you have your subglacial microbes. Within the ice cover where the glacial ice forms, you also have a variety of microbes that are trapped within the ice, right? And they just fall with precipitation. Are these microbes viable, or are they just frozen in time? It's still an outstanding question. Then you have surface microbial communities that either occupy subglacial or superglacial streams that form on the surface, and many of these are photosynthetic group. And then you have microbes that like the grounding zone area, and they can interact with the marine system and maybe even grab some of the nutrients that are coming out of the subglacial setting. And of course, the saturated sediments that go deep below the ice can have microbes that inhabit this region as well. And so here are just a few of some of my favorite reasons why I think everyone should care about subglacial microbes, but I hope it highlights the diversity of areas that you could study when you look at glacial microbiology. So the first is bioalbedo, and this is microbes that occupy the surface. Often they're heavily pigmented, either to protect themselves against the sun or as part of their photosynthetic machinery. And these microbes can influence the rate of melting of glaciers, right? Because a pigmented microbe will absorb radiation differently than fresh snow. Glaciers are also serve as cold storage. And so as you go deeper in glacial ice, you go back in time, and there you find stories of our past locked in the genomes or other attributes of these microbes that are within the ice. Some microbes are below the glaciers in particular are eating rocks to stay alive, right? So these are rock-eating microbes, and their waste or metabolic byproducts can be discharged along some of those flow paths and discharged into the ocean. And this can influence global biogeochemical cycles, right? Especially if these rock-eating microbes are liberating limiting nutrients such as iron or silica to the ocean. What we see in these subglacial environments is that these microbes are actually alive, and that means they're metabolically active. And so they're consuming organic matter or this bedrock material, and they're releasing CO2 or methane or dimethyl sulfide or other types of substrates. And in the case of CO2, methane, and dimethyl sulfide, these are greenhouse gases. And so the knowing about the extent of these microbes and their metabolic rates is really important for understanding how they might impact global climate. There's also this notion of bioprospecting. I mentioned earlier that Antarctica was a microbial continent. So it's kind of like this large untapped resource of organisms that are living in extreme cold and darkness, and their molecular machinery could produce molecules that are important for humans that might be novel therapeutics, novel antibiotics, or potentially really important industrial products that can help us operate more efficiently in our changing world. So I really am excited to explore some of the microorganisms we're able to tease out of these environments. Okay, so back to my figure that kind of orients us on the scale. So I'm gonna try to tell 3 quick stories about how we operate with geophysicists, engineers, and then what we can actually do once we get the samples. So I mentioned that geophysics can reveal where potential habitat might be. One example where we use this tool was a tool called SkyTem. And this is a helicopter-borne survey that flies around this big sling load that is an electromagnetic system that will map resistivity in the ground beneath it. And this can reveal salty systems or brines. And so we use this to fly over a glacier in the McMurdo Dry Valleys called Taylor Glacier. And where there was glacial ice, it was very resistive, and that's this purple color. This is the data that the SkyTem produces. And where there was actual saturated sediments, you found this bluish brine, bluish color, which is indicative of brine. So this helicopter survey helped us find our needle in the haystack by identifying where the brine was below this thick ice sheet. We then went there to drill. We kind of cheated a little here. We knew there was brine that came out. This is a feature known as Blood Falls, and the salty red iron-rich water comes out of the glacier. And so we wanted to try to sample it before it came out. And so once we used SkyTem to detect where that aquifer was located, we used a novel drill to melt into the glacier and collect samples cleanly for microbiological analysis. And for this work, we collaborated with engineers. In this case, these were engineers funded by the German Space Agency, and they designed a tool called IceMole, which could melt through the glacier and go in and then use this novel sampling system to suck up sample and bring it back to us to work on for microbiology. So after all of that, when we finally get a sample, we can bring it back to the lab in our clean hoods and work in a very sterile setting to examine what microbes might be present in these samples. So here you see the DNA of microbial cells lit up under the fluorescent microscope. We can then start to target these cells by creating media that will coax them to grow. So for these subglacial microbes, we tried to give them iron substrates so that it would mimic the subglacial environment. And then we could take more fancy pictures with the electron microscope and reveal some of the morphology of these cells. You can see they're really quite attractive, not just these squiggles that you see with the fluorescent microscope, but these beautiful flagella. And then we can challenge them to different conditions that mimic their environment and see what they prefer and maybe what they do when they grow under their conditions. Do they grow different compounds when they're challenged at different temperatures? And while we see fastest growth at warmer temperatures with this particular organism, a strain of Shewanella, it actually grows more optimally at lower temperatures. So fastest growth isn't always optimal. We can then see if what we see in culture or in its phenotype matches what we see in its genome. And so we have a lot of tools these days that provide us with the ability to sequence genomes even on the bench in the average lab, right? And so here we have an undergrad, Bruce Bowles, and he is using this small telephone-sized, even smaller than a cell phone, size sequencer called the Oxford Nanopore MinION, and he used that to generate the genome sequence of the Shewanella I showed you. And then he can see if it has the traits that allow it to survive in icy environments and if it matches what we see in culture. If you can do some of this work in Antarctica, why not try to look for life on places like Europa or Mars? You can use some of these tools. You'll still need geophysics to help you identify where to target. You'll need engineers to create tools to go there and collect samples. And then if you are so fortunate to get your hands on some of those samples, you'll need a variety of collaborations to interrogate them. But a task like this would require even many more hands than even just to go to Antarctica. So with that, I'd like to thank you for your attention, and I'd like to thank my lab. We call ourselves Ice Microbes. We're at the University of Tennessee, which is in Knoxville. Because of these diverse collaborations, we have questions that span a variety of topics, from how microbes survive in these environments, their adaptations, to astrobiology. We are part of the University of Tennessee Microbiology Department. It's in beautiful Knoxville with the Smoky Mountains just about 40 minutes away. We have a vibrant department that includes not just folks who study extremophiles, but folks who are interested in pathogenesis and genetics and virology and host symbiont relationships. So we really have a diverse department. I encourage you to check us out. There's a lot of outdoor activity, mountain biking. This is actually a whole mountain biking trail network here. And we also have research experience for undergraduate programs, including one that's associated with the National Institute for Health. And I leave the information here if it's something that you're interested in pursuing, and you can come to Knoxville and check out what we have to offer. So thank you.
Thanks again, everyone, for joining us for the recorded session. I hope we'll see you now live.